All right, hello everybody. I just want to welcome you all to the Lab 4 overview. This should be pretty brief and straightforward. Lab 4 is a carbohydrate lab. Basically, what we would do if we were in person is we would give you various solutions, and not only for carbohydrates, but normally we do a triglyceride test using vegetable oil. We also do a protein test using some chemicals as well as just some everyday solutions. But this lab just focuses on carbohydrates. Now we talk about carbohydrates, and if you look here, the objectives of this lab is to be able to explain what an indicator or indicator solution is and explain why each one is used in your labs. Include Benedict's reagent and Lugol's iodine. I'll discuss this as we go, and I'll talk about how these solutions we will first use to set up what we call positive control test and negative control test. So part of the other objective is being able to differentiate between what do we mean by a positive control test versus a negative control test, and how do those two relate to your experimental test. And then last, explain how to test for monosaccharides, which are simple carbohydrates, and polysaccharides, which are complex carbohydrates, and how to interpret these tests. So these all three objectives are rolled together in the end as you go through this lab. And you'll hear me say some of the times you'll see titles instead of simple carbohydrates, you'll see simple sugars. Okay, in either case, one and the same, and they're talking about monosaccharides. In this lab, the monosaccharide will be glucose. You'll also see in the lab complex carbohydrates or complex sugars, and what they're talking about is polysaccharides. And in this lab, you will be testing for the polysaccharide starch. So when it comes right down to it, there'll be one type of monosaccharide glucose, one type of polysaccharide starch that you'll be testing for in solution. So basically, with the lab, like I said, usually involves testing for a lot more than carbohydrates. So I talk to students about this lab being our organic compounds lab. DNA and nucleic acids and all that, we usually do that separately in a different lab, but we're not doing it online. So as I talked about, we talked about carbohydrates, and we talked about how carbohydrates, along with proteins and nucleic acids, are formed by building blocks called monomers. But instead of saying carbohydrate monomers, I talked about how we say monosaccharides. And down here, they are off to the right here, showing you the two types of monosaccharides in lecture you needed to know. Well, I give them to you here in lab, glucose and fructose. In today's lab, when you test a solution for simple sugars or simple, what we call it, carbohydrates, you'll be testing solutions for glucose. Okay, but we also talk about complex sugars or complex carbohydrates, in which case these are polysaccharides. And we talked about how polysaccharides consist of cellulose, starch, and glycogen. Well, in this lab, you'll be specifically testing for starch, that polysaccharide, which is shown down here also on these two. Okay, so the lab's very focused. It's not as broad as what we covered in lecture, okay? Now, first objective was becoming familiar with what we call color indicators. I call them chemical color indicators or chemical indicators because these are chemical reagents, solutions, but they're chemical reagents that allow you to test for either glucose or they allow you to test for starch. So normally what would happen is you would be given a tray by our lab tech, Joel, out at Hayes campus if you were taking it out there. You would have a tray and you would have various solutions on there that you were going to test for the presence of glucose in that solution or the presence of starch. But also on that tray you would have two bottles. They're a different color. They're usually a darker brown colored bottle. And one would say Benedict's reagent and the other one would say Lugol's iodine. Okay. These two, okay, are solutions I call chemical color indicators or chemical indicators because in the case of Benedict's, you'll see a dark brown bottle, but when you're transferring Benedict's to any solution you want to test for glucose, you'll see the Benedict's is actually blue, okay? And then for Lugol's, the bottle itself is usually a dark brown bottle, but anytime you want to test a solution for starch, you use Lugol's, and it has actually a natural yellow color. Now, the way it works is whenever you want to test a solution for glucose in this lab, you'll use this chemical color indicator, Benedict's reagent. When you add it to a solution, okay, that contains glucose, initially, if it's a clear color solution, initially it's going to turn the solution blue. The way it works for Benedict's is the only way it will react with any glucose in a solution, okay? The only way it'll react with any glucose that's present in a solution is if you place it in boiling water. This chemical here has to be heated in its solution form, has to be heated for it to cause it to react with glucose. And if glucose is in that solution, the Benedict's will cause the solution to turn a yellow, yellow-orange, or more of an orange color. It depends on the amount of glucose. And it's a cloudy color. It's what we call a yellow-orange precipitate. It's a very cloudy color. So you could tell. However, if you ever add Benedict's to a solution that doesn't have glucose in it, after you place that sample, that mixture of the solution you're testing for glucose that doesn't have it, and Benedict's reagent in boiling water for 1 to 2 minutes, the solution will stay the color of Benedict's blue. So you know it's negative. So we'll get into the positive and negative control test. Likewise with Lugol's iodine here, it's a much simpler test. First and foremost, if you read here, there's no mention of boiling like there is up here for Benedict's. Lugol's is real quick. But whenever you want to test a solution for the presence of starch in that solution, you take a little bit of the solution, transfer it to a porcelain dish or a test tube, add a little bit of Lugol's iodine to it, okay? If there's no glucose in the solution, and the solution's a clear solution, it'll just turn it yellow. Some solutions we test for both using Lugol's aren't a clear color, so you're not going to see a blue or a yellow, but it doesn't matter. You will see the positive color for sure if it's positive. But in either case, if you add Lugol's to a clear color solution testing for starch, if there's no starch, it'll just turn the solution yellow. But if you add Lugol's iodine to a solution that does contain starch, okay, it'll immediately turn it to a blue or blue-black or black color. It depends on how much starch is present. The more starch is present, the darker it is or the blacker it is. Okay, so these are the chemical color indicators. This is the first objective, and this is what you'll be focusing on for your test. So you have to know how these two chemical color indicators work in order to understand the rest of the lab and to be able to answer your questions. The next part of your lab, and this is the next objective, is understanding the 3 types of tests that are run for the lab. What we normally do is we have students take Benedict's reagent and set up 2 tests, a positive control test and a negative control test. And then we have them take Lugol's, and we have them set up a positive control test and a negative control test. And your lab is set up that way to walk you through. And in your lab, they'll actually show you the results from the positive and negative control test. But the purpose of the positive control test, simply put, I'm not going to read through this verbatim. The purpose of the positive control test, okay, is to demonstrate to you, hey, this is what it looks like whenever we add Benedict's to a solution that contains glucose, so that you know what we mean by yellow, yellow-orange, what we mean by a cloudy precipitate that has a yellow-orange color to it, okay? Likewise, when we talk about a positive control test for starch, what we're talking about is showing you how Lugol's, when it reacts with starch in solution, what we mean by it'll turn the solution a blue, black, or a black color. Okay, so in the lab, the positive control would be a 1% starch solution. We would give you a 1% starch solution. You would transfer a couple of drops to what we call a porcelain dish, which has these little wells in it. You would add a couple of drops of Lugol's, and you would get a positive result, which would be that bluish black color. So that way you know, okay, when I test solutions that I don't know if they have starch or not, I can compare them to this positive control result, so I know whether or not they're positive. Likewise over here, what I didn't say for Benedict's, to backtrack, is normally we give students a diluted form of Caro syrup. Okay, this is a Caro syrup you can find in your cereal aisle next to all the other syrups, and the Caro syrup that's used to make pecan pies, unless you don't make pecan pies, never mind. Okay, but the Caro syrup, it's a sugar solution. It's a glucose solution. It's just like sodas. Okay, if you're drinking a soda that is not a diet soda, it's going to have glucose in it, but we say sugar. Okay, but it's glucose in it. All right, so if you look there for Caro syrup, okay, likewise, when you add Benedict's reagent, but you place it in boiling water, you should get a positive result. That way, when you're testing, so you don't know if they have glucose in them, okay, you'll be able to compare the results when you test it with Benedict's to this positive control. Compare them based on color. Likewise, we do negative controls, just so you see what I mean by if you add Benedict's reagent to a solution that doesn't have glucose, okay, well, it's just going to tune the solution the color of Benedict's, which is blue if it's a clear solution. Likewise, if you add Lugol's iodine to a solution that doesn't have starch, it's going to turn the solution yellow, which is the color of Lugol's. Okay, now sometimes, as I said, that can be obscured if you're using a solution like apple juice or something, which has a brown color. Yeah, it's harder to see that blue color caused by Benedict's or that dark yellow color caused by Lugol's, but the thing is, you would take into consideration both your positive control test and your negative to help you, okay, deduce or come to the conclusion of, okay, the sample that I didn't know if it had glucose or starch, let me compare it to the positive or negative. It should match one of the two or not match one of the two to let me know, okay? So in this lab, Okay, what we do is when you read through the lab and you get to the actual experiment, it'll tell you, hey, you're going to set up your positive and negative control tests for glucose. You're going to use Caro syrup, which is a glucose solution, because it should test positive for glucose when you add Benedict's and place it in boiling water. But for the negative control, you just simply use DI water, deionized water, DH2O. You would add Benedict's to that, place it in boiling water. After 1 to 2 minutes, the water should stay the color the Benedict's turned it, blue. And you'll see that in your lab. Likewise, when you're testing for starch, it parallels the same process. You transfer a couple of drops of 1% starch solution to a little well in a porcelain dish. You transfer a couple of drops of DI water or deionized water to another well, a couple of drops, and then you add Lugol's in both, and you'll see what a negative result looks like when you add the Lugol's to the water versus when you add it to the 1% starch solution with a positive result. This is given to you in the lab. They not only tell you, they show you visuals of what I am talking about, okay? Now, and keep in mind when you're looking at that, when you're looking at those visuals, when you actually go to the lab experiment, pull up this slide 5. This is slide 5. Look at what I gave you for Benedict's. Look at what I gave you for Lugol's as far as negative, positive results, okay? And use it when you get into the actual experiment for the controls. Then it'll ask you to fill out a table based on your positive and negative control results for glucose and your positive and negative control results for starch. Okay, so you'll need the slide, especially slide 5, along with the tables and the procedure it gives you, along with the pictures it gives you to fill in the table. If you just take the time to read through everything thoroughly and listen to this overview, okay, somewhere in there, you should be able to see, hey, this isn't hard. Okay, I just have to pay attention to what I'm doing. Then what it'll do is it'll take you into the experiment test. It says here, when you test the solution that you don't already know if it contains a compound you're interested in, such as a simple sugar such as glucose or starch. I forgot to change simple sugar to glucose, so this it says glucose and I'll edit it, or starch. Then this is called the experimental test, okay? Your results allow you to find out if the solution does or does not contain the compound when compared to the positive and negative control test. So when you finish going through the information they give you and the results they give you and filling out the table for the positive and negative control test for both using Benedict's for glucose and Lugol's for starch, you're going to keep referring back when you look at the solutions that you're going to be testing in the experimental part, because you have several solutions you're going to test. And I'll show you that when I pull up the PDF. Or actually, I'll show it to you now. I forgot I put it on here. Okay. And I just went over this to make sure I don't miss anything. So you're going to see a table like this, okay, in your lab. And these are the experimental test solutions. So up here it tells you these are the solutions that your experimental test. And you have all these solutions. And it tells you, hey, you're going to test the solutions for glucose. Well, any solution you're testing for glucose, you're going to use Benedict's. And then it tells you over here, you're going to test for starch. I'm sorry, I meant to underline it here. You're going to test for starch. So any solution you're going to test for starch, you're going to test using Lugol's. And the way it works is whatever procedure you followed when you were testing your positive and negative controls for Benedict's and when you were testing your positive and negative controls for starch, you follow the same steps as far as how many drops of each of these solutions will I use, how many drops of Benedict's will I use, how many drops of Lugol's will I add to the sample that I transfer. It's the same procedure. You're just replacing the positive and negative control solutions for your experimental solutions. And then you record your results. Well, that's done for you in the lab. It's just up to you to be able to interpret what the results are so you can fill out the table in your lab. So the lab itself is pretty straightforward. Answer all questions. There are some questions embedded under the procedures. So make sure you read word for word so you don't miss. There's a couple of them that are question marks. Okay, and then the table information stands out. You know to fill in all the boxes. I got a lot of questions about the salt lab, lab 2. Do I need to fill in all the boxes? I covered all that in the interview. So listen in this overview. I'm sorry, in this overview. So it's all there, and I know most of you heard me. Okay, so it's up to y'all to listen to this overview. Of course, there are going to be those that don't listen to it, so it doesn't make any sense for me to keep repeating it. Okay, let me pull this up. This is a PDF, okay, that you have. It starts off with the objectives, and then you'll see it initially goes into the testing, which is the first objective, the chemical color indicators, okay, Benedict's and Lugol's. What it'll do then is it'll transition into the positive controls Then it'll move into the negative controls, and then it'll move into the experimental test. Then what it'll do is it'll have you here set up your positive control test for glucose using Benedict's reagent, and it'll give you the results for the positive control, the plus, and the negative control here. And then all you're going to do is read the steps, answer any questions, and then fill in the table. Then it's going to take you into the test for starch using Lugol's. It's going to take you to the steps, and again, it gives you the results for the positive control and for the negative control. And then all you're going to do is reading through all these steps and looking at your results. You're going to fill out the table. Okay. Then it's going to get into your experimental testing. It gives you all the solutions, which are the ones here I showed you. And in this part right here, this is where you're going to be testing for glucose. You have to know which of the two solutions do I use to test for glucose. Well, it's Benedict's or Lugol's. 50-50 chance you get it right, right? You should get it right, no problem. And then over here would be Lugol's, okay, versus Benedict's here. So you'll fill in the table, but you're given what the results should look like. This right here is for the Benedict's testing for glucose, or the glucose test. This here is a starch test using Lugol's. So all you do is analyze these, take a step back, and fill in the table, okay? So there's two tables because, I'm sorry, I forgot to mention this. There's two tables because this table here wants you to predict whether the solution has glucose, whether it has starch, okay? This table here, you're going to fill in based on these figures here, okay? So these figures here, I'm sorry, I got ahead of myself. These figures here, the results, you're going to put on in the table when they show it to you a second time. But read through it word for word because there's still questions you have to answer. Then it's going to talk to you about diet soda. And I just told you about diet soda earlier in this overview. So it's up to you to listen to what I said. And when I slipped it in there about diet versus non-diet soda to help you answer these questions, okay? And then you're just going to answer the questions on the last page here. So it's much the same. Most of y'all, if not all of y'all, been doing a good job of just inserting your answers either into the PDF or Word. Remember, it's your job to make sure that all I have to do is go into Blackboard assignments, select your lab or your homework, open up the file, and I can grade it right there. That also gives me a chance to give you feedback a lot easier, okay? Most of you have done well, knock on wood. Keep doing what you're doing. The rest of you that aren't on board yet, got to catch up, everybody. Catch up, okay? All right, then y'all take care. That's it for the lab 4 overview. Bye.